Good, a very warm welcome back to our session, to the anatomy session, and we continue with Professor Karl Zilles, and he opens up a new perspective of relating structural and functional aspects. And when we speak about receptors and neurotransmitters, we are perhaps in a situation where we cannot easily say this is structure or this is function in an old-fashioned way, and we will learn in this talk more about how the receptors of neurotransmitter systems are related to resting state. So, Karl, thank you for coming very much.
So, next slide. Oh, yeah, I can write here. My question is, why are neurotransmitter receptors important for understanding the neurobiology of the default mode? I think the default mode network is one of the exciting concepts in recent neuroimaging. And we know a lot about the metabolism of the default mode network and how this network behaves during task-related stimulation or during resting state. But we do not know why. What is the underlying molecular mechanism? Or other perspective, we have heard about macroscale, mesoscale, microscale up to now. Now I try to bridge nanoscale receptors with mesoscale, the concept of resting state systems and default mode network. The human brain is 2% of body's weight, but accounts for 20% of its resting state metabolism. And movements largely contributes to that. And in some papers, the people say up to 60, 70, 80% of the metabolism, of the energy metabolism, of oxygen metabolism is used by channels. And the receptors are subdividable in such receptors which have channels, other receptors which control channels, but via an indirect way over G proteins. It was proposed that glutamatergic synapses, being major users of metabolic energy, and that the activity of inhibitory GABA neurons may lead to less postsynaptic energy use than the activity of excitatory neurons. This sounds a little bit simple. Excitation means, according to this, energy use. Inhibition means saving of energy, and the contrary. So it would be interesting to test whether this is true or not. The argument is that inhibitory synapses need less energy because chloride ions move down a smaller electrochemical gradient and at inhibitory synapses than sodium ions do at excitatory synapses. And I think my talk will also be an example that our best knowledge of single molecules at the single molecular level does not allow to predict the behavior of microcircuitry or cortical areas, what is of interesting for us. So Rainer has shown how macro predictions must be corrected if you look at high resolution into areas. And I have to say that biochemical nano predictions have to be corrected if we look at the level of microcircuitry or of cortical areas. So at the end, we must say the role of transmitter receptors as key molecules in such processes of information processing has, however, never been studied at the neural systems level, for instance, at the level of the default mode network. During the last years, I have given talks here in the educational course about transmitter receptors. Therefore, I should avoid to do it again and to speak again about the transmitter receptors and their mapping in detail, but a few basics I have to mention before I come to the default mode network. Here you see the scheme of a synapse, and the synapse has postsynaptic receptors, presynaptic receptors, and transmitters. The transmitters are released by the presynaptic axonal bouton, and they bind to receptors, either postsynaptic receptors or presynaptic receptors. And these receptors are expressed by neurons. And a single neuron expresses many different receptors for many different transmitters. That means that a GABAergic neuron does not only produce GABA and may have presynaptic GABA autoreceptors, it expresses also glutamate, cholinergic, adrenergic receptors, many others. And the pyramidal cell, scheme is here, has receptors for glutamate, GABA, acetylcholine, dopamine, noradrenaline, and serotonin. In detail, we do not know presently all the details of the afferents to each neuronal type. If we want to understand in the future the microcircuitry, then we have to do that. And I will show you a few examples. Receptor classification. Based on pharmacological characteristics, receptors are classified by the neurotransmitter which binds to this receptor. This would be a pharmacological classification. But also other classifications are possible. For instance, one based on outcome. The receptors can be excitatory or inhibitory. In this respect, receptors are very, very clear. They are either excitatory or inhibitory, which is in sharp contrast to the transmitters. Many people, also many textbooks, speak of inhibitory transmitter or excitatory transmitter. This is strictly wrong. And transmitter can be excitatory or inhibitory, depending on the receptor to which this transmitter actually binds. Therefore, I prefer to restrict the terms excitatory and inhibitory to the receptor and not to the transmitter. Excitatory receptors are, for instance, the glutamatergic AMPA and NMDA and kainate receptors, but also many other receptors. Inhibitory receptors are the GABAergic GABA-A receptors, but also many other receptors are inhibitory. And the third types of receptor classification is based on the mechanism of action. A receptor can have an integrated ion channel, or it cannot have such an ion channel, but is connected to G proteins. And on indirect way, these G proteins can, amongst other functions, influence ion channels which are not bound to receptors or which are bound to receptors. So at the end, we must say that the transmitter receptors are influencing ion channels either directly or indirectly. And the introductory remark was that much of the energy metabolism is used for keeping the ion fluxes or for keeping the membrane potential after excitation or inhibition again at the normal level. No, I have included in the study, which I would like to show you, not all possible receptors in the brain. These are more than 100 different types and subtypes. I have selected all classical transmitter systems like glutamate, GABA, acetylcholine, noradrenaline, serotonin, and dopamine. And from each of these transmitter systems, I have selected the most abundant receptors in the cerebral cortex of the human brain. And this is shown here. And these receptors are either ionotropic, they have an ion channel integrated, or they are metabotropic. They act via G proteins. They are either excitatory, in red, or inhibitory, in light blue here. Now, let's go to the default network. The first challenge was to compare the different depictions of the default mode network in the literature. The default network seems to be well defined at the resolution of PET or standard MRI. But an anatomist is not completely satisfied. He wants to know the regions. And therefore, I have selected two of the most frequently cited schemes of the default mode network. One published by Buckner et al., and the other one by Ragley and collaborators. And when you map their default mode network to the U-Brain Atlas Catherine has shown, or to the Brodmann map, then you can define a number of cortical areas which are cytoarchitectonically defined, which are relatively precise in their extension and localization. And that's what I have done. I have measured the receptor densities of all receptors I have shown you, of all these 15, I think, different receptor types, in areas which are clearly demarcated by cytoarchitectonic criteria in 9 different hemispheres. To give you an impression how the receptor density is distributed in coronal sections of post-mortem human brains, you can see on the left side a frontal section, on the right side also a frontal section, more in the central region. And what is shown here is the distribution of the glutamatergic AMPA receptor. The red numbers define default mode areas, and the white letters or numbers define non-default mode areas. And you will get the impression, it's only to introduce you, the impression that the kainate receptors show a somewhat higher density in the default mode areas than in the non-default mode areas. The kainate receptors are most abundant in the deeper cortical layers, and therefore this increase of kainate receptors is seen mainly in the deeper cortical areas. For instance, area 36 and the entorhinal cortex. Entorhinal cortex is EC, area 36. These areas have a high density. Also, the inferior temporal lobe or gyrus, area 20, but area 22 and the sensory A1 area, this is Heschel's gyrus, have a very low kainate receptor density. So the red numbers and letters depict the areas of the default mode network, and the white numbers and letters of non-default mode network. When we compare the functional definition of the default mode network after Buchner and Rackler and Mintum, that's shown here. These areas are default mode network areas with the distribution of AMPA receptors. And here we have measured the absolute density of the receptors in femtomole per milligram protein. You will not see very much a general tendency, except perhaps this low density of the kainate receptor in the motor cortex, area 4, area 6, and parts of area 8, and the very low density also in the primary visual and the primary somatosensory cortex and the primary auditory cortex. Therefore, I have simplified this and mapped only high receptor densities in red and low receptor densities in blue. And immediately you see that the prefrontal part here of the default mode network lights up, that the temporal part of the prefrontal network lights up, but also parts which are definitely not default mode network have a very high AMPA receptor density. My conclusion, a single receptor, and I will show you a few more examples, a single receptor does not define default mode network or resting state network. Let's go to the next receptor. The GABA A receptor, inhibitory receptor, the GABA B receptor, and the benzodiazepine binding sites of the GABA A receptor. And again, here, you seem to have high density in part of the default mode network here, and also on the cingulate and medial prefrontal cortex, but you have also high densities in areas which are definitely not part of the default mode network. And therefore, also here holds true, a single receptor does not define the default mode network. Then let's go to the cholinergic receptors. Also here we have again in the frontal region high densities. We have, but here in the auditory high densities, which is not part of the default mode network. Here in the entorhinal cortex and in the ectorhinal cortex, we have again high density, which is part of the network. So here, for the third time, a single receptor does not define the default mode network. Now let's go to the next level of analysis. We can ask a question, if not a single receptor defines a default mode network, does eventually all the receptors together interact in an area in such a way that a specific balance between the receptors is reached, and that's what I call a receptor fingerprint, a multi-receptor fingerprint. And now we have done this for all areas. For instance, the S1 area, the primary somatosensory cortex, the primary visual cortex, the primary auditory cortex. All these three areas are sensory areas and not part of the default mode network. And here around this polar plot, you have the absolute densities of the different receptors, of the glutamate receptors, AMPA and MDR kinase, of the GABA receptors, GABA A, GABA B, GABA A, benzodiazepine binding sites, of the cholinergic receptors, muscarinic M1, M2, M3, nicotinic alpha-4, beta-2, adrenergic alpha-1, alpha-2, serotonin receptor 5-HT1A, 5-HT2, and dopamine D1. And here is the absolute density. And when you connect the different data of the different receptors, then you will get this figure, this area. This is the receptor fingerprint. And the size and the shape of the fingerprint tells you something about the sum of receptor expressions. Are there many receptors in the area? That means also, are there many synapses in the area? And it shows you the balance between the different receptor types. When you look at these receptor fingerprints, then the fingerprints for the different primary functions, somatosensory, visual, auditory, the shapes are very different. And in fact, we can trace by the fingerprint the auditory system, all cortical areas which belong to that system. We can trace the visual system, we can trace the somatosensory system. This would be something like a resting state approach. And look here, very different. The prefrontal area 10 medial, that's PG2, what Catherine has shown. The area PG, which Svenja has shown in her work on the inferior parietal cortex, and the area P24 seem to be relatively similar compared with this. Now we have to make a more strict evaluation of the similarity. Are they really similar and different from the other areas? And that's here. That is a principal component analysis of the receptor fingerprints. So this study is multimodal because it's not a single receptor, it's the balance between all the receptors. And what you see in the red, these are all areas which are classified as default mode areas. And they are more similar to each other than the other areas in yellow or in green or in white, which are other systems of the brain. So the default mode system has a very specific receptor constellation, which is completely different from all the other areas and which is clearly separated by the principal component analysis. And we can also take a hierarchical cluster analysis of the default mode areas shown on the right and on the default mode areas based on the receptor fingerprints. And what you see here as red, marked in red, all these areas are areas of the default mode. Hippocampus, prefrontal cortex, anterior and posterior cingulate cortex, and areas in the inferior parietal lobule and the precuneus. This is the default mode system, and this, you see, is principally on a different branch of the cluster tree than all the other systems. Yellow, visual; green, auditory, that's language, broca, and that's motor. So also in this type of analysis, the default mode system is separated from the rest of the cortex. When we ask the question, has the default mode system more or less receptors than the other systems? The question is quite clear: yes, the default mode systems has much more receptors and therefore probably much more synapses than all the other cortical regions. And surprisingly, to this separation between default mode and the rest of the cortical systems, not AMPA, the main excitatory receptor, and not GABA-A, the main inhibitory receptor, are most important. It's kinase 5-HT2, 5-HT1A, and GABA and AMPA is in the second half of the most important receptors. So that argues that the interaction of receptors in the microcircuitry of the default mode areas is even more important than the occurrence of the most impressive excitatory and inhibitory receptors. Now, after having this data, we can do the last step. We can directly compare the oxygen consumption with the receptors. And I can tell you that this delivers very, very surprising results. Here you see in these regression curves, on the left side, always the data from the paper of Haider et al. About the cerebral metabolic rate of oxygen consumption. And on the right side, you will see always the same data which Marcus Reichler and collaborators has given to me to compare it with the receptors. And what you see, can we go back? Back once. Ah, yeah, it's all right. What is most surprising is the negative correlation of all cortical areas, not only of the default mode areas, which are shown in red as red dots, but also of the non-default mode areas, the black dots. That means the more GABA receptors, the more excitatory GABA receptors in a cortical area occur, the less is the oxygen consumption. This is absolutely in contrast to all speculations which have been done about AMPA receptors. But it does not exclude that a single AMPA receptor needs more energy than a single other receptor. It says only that at the level of systems, of microcircuitry, of cortical areas, AMPA receptors are most densely present in cortical areas which have the lowest extraction rate of oxygen. Then we come to the next. Is this only for AMPA receptors? No, it's also for adrenergic alpha-1 receptors. They show the same. In the default mode areas and the non-default mode areas, the oxygen extraction goes down with increasing receptor density. And now, for the first time, separation between default mode and non-default mode networks. Here, for the 5-HT1 receptor, irrespective of the metabolic data from one group or another group, you see that the oxygen extraction rate has a very steep slope in the default mode network and a lesser deep slope in the non-default mode network. That's a clear difference in metabolic behavior of such receptors in default or non-default mode networks. Next. Is this always the case, this negative correlation? Answer, no. Here you see the alpha-4 beta-2 nicotinic receptor, and you can clearly see that in the default mode network, the oxygen rate extraction increases with increasing receptor density. And the GABA-A receptor density increases and causes increasing oxygen extraction. These results were really surprising because they are in contrast to the results you get on the single molecular level if you study only the receptor isolated from all other receptors, from all connectivities in a microsystem. Therefore, we come to the following summary. The cytoarchitectonically defined areas and regions of the default mode network differ from all other brain regions by their receptor fingerprints. That was the principal component analysis and the hierarchical cluster analysis. The hierarchical cluster analysis and the principal component analysis of receptor fingerprints show clear segregation of the DMN from visual, auditory, and motor networks. Second, the densities of receptors of excitatory, glutamatergic, AMPA, and noradrenergic alpha-1 receptors, as well as of the inhibitory serotoninergic 5-HT1A receptors, are negatively correlated with the cerebral metabolic rate of oxygen in all brain regions, in all NMDA, DMN, and non-DNM brain regions. The density of excitatory glutamatergic kinase, nicotinic alpha-4 beta-2, and dopaminergic D1 receptors, as well as inhibitory GABAergic GABA-A and muscarinic M2 receptors, are positively correlated with the cerebral metabolic rates of oxygen in default mode network areas. And the conclusion is the characteristic metabolic functional feature of the default mode network is probably caused by the interaction of various excitatory and inhibitory receptors and their actual cellular localization in the microcircuitry of different brain regions of the default mode network. I think that this shows that the receptor analysis can really bridge the mesoscopic finding, interesting finding of the default mode network with all its functional implications, with the molecular basis of this separation, exemplary separation of cortical areas in the human brain. Thank you very much.
Thank you, Karl, for these exciting new insights. Oh, yeah, okay, so can you start and then Rainer?